Thanks, Martin and Tamby. Now in Part 3, I will briefly describe the plans for transition to Universal Prospective WGS. A separate presentation covering the details of how Universal Prospective WGS will be implemented will be made available in the future. Universal Prospective WGS begins in 2018, and WGS of isolates from all new culture-confirmed cases of TB will be performed. But GENType will continue to be analyzed during an initial 3-year transition period, which will be 2018 through 2020. During this time, GENType will continue to be reported in TB GIMS, and cluster alerts will be based on GENType. In 2021, WGS will become the standard method for TB genotyping. WGS data will be used for 2 separate analyses to examine transmission: wgMLST, which is whole genome multi-locus sequence typing, and wgSNP, which is whole genome single nucleotide polymorphism analysis. And I will explain these 2 analyses in more detail in the following slides. Although wgMLST and wgSNP both use WGS data, they differ because wgMLST is a scheme that will be applied to all isolates to compare the genomic sequences of the isolates and assign them to a wgMLSType that can be used for cluster alerting. The wgMLSType is a standard naming scheme that will be a short string of numbers similar to a GENType. On the other hand, we use wgSNP to examine the genetic relationships among isolates in a cluster in more detail, and the cluster can be based either on GENType or wgMLSType, and the output is a phylogenetic tree that diagrams these genetic relationships. This is what the TB genotyping methods and data flow will look like for the 3-year transition period. During this time, we will be performing whole genome sequencing as well as conventional genotyping with spoligotype and 24-locus miru for each isolate. GENType will continue to be analyzed and reported in TB GIMS, and GENType will be used for cluster alerting. The WGS data will be used for wgMLST and wgSNP analysis. However, wgMLSType won't be displayed in TB GIMS until after the initial 3-year overlap period in 2021. This will allow for the necessary time to adapt algorithms for using wgMLST to define clusters. For clusters that alert during this time based on GENType, wgSNP analysis will be performed to assess the potential of recent transmission, and this information will be communicated to the state program. For clusters that have had previous retrospective WGS performed, any new isolates in the cluster will be added to the analysis, and the updated results will be reported to the state program. In 2021, WGS will become the standard method performed to identify TB clusters, and wgMLSType will be used to generate cluster alerts. For clusters that alert, the genetic similarities among the clustered isolates can be examined in more detail with wgSNP analysis. We would like to thank the TB Outbreak Prevention and Control Section at the California Department of Public Health, the Applied Research Team and Molecular Epidemiology Activity at DTBE, the Microbial Diseases Lab at the California Department of Public Health, and the Association of Public Health Laboratories.